So I'm going to start not with my original plan, but just for a little bit of continuation of discussion of Larry Sandler's legacy. And so I'm an example of one of his scientific grandchildren, as is Leo, and I'm sure many, many others. And actually, I searched on the web. I was looking for a pedigree. I didn't find one, but I thought there is one out there somewhere, and I would love to have it if someone could send it to me. So I got Larry Sandler's legacy through Bruce Baker, who Barry mentioned, passed away recently. And I have to say that everything that Barry said about Larry, I mean, Bruce also said all of that. He really revered Larry. And so one of the things I got through my inheritance from my scientific grandfather was obviously a love of genetics. And it's just wonderful to spend a whole day, hopefully, hearing about all sorts of genetics and that legacy. Also, working as a team. And in Bruce's lab, we always aspired to this. We didn't end up doing what Larry's group and Dan Lindsley's group did together, which was this amazing segmental aneuploidy paper looking at all the dosage effects of across the Drosophila genome, which was just such a foundational paper, not just in Drosophila genetics, but in genetics in general. And we aspired to doing something like that. We never did. But I did follow up in the sense of working on gene dosage, the importance of gene dosage in genetics. And so what I did with Bruce Baker, what Bruce Baker and I did was to study gene dosage in the context of sex determination. So as you all know, being aneuploid or having multiple or having a loss of a chromosome is quite deadly in most organisms, except for the sex chromosome. And that is that males, there's a very important X chromosome. Females have two. It's also important in males, but males only have one. So how do they survive? And so what we found, I'm just going to summarize a little bit, of what we found, which was that there is a specialized male-specific, X chromosome-specific modification and whole system of proteins. And really, it's a ribonuclear protein complex that binds that X chromosome. These are polytene chromosomes showing the exquisite specificity of this machinery. And what it's doing is helping the male X chromosome express twice as well, each gene twice as well as female X chromosome, so that in the end, 2 X's equals the single male X. So I'm just going to summarize what's sort of the consensus in the field from most of that work, which is, first of all, the important protein components were named for their mutant phenotype, as is the habit of Drosophila geneticists, male-specific lethal, or MSL proteins. The genes were all found by John Lucchese and Kagawa Oishi. And what it turned out that they're doing is they're putting on a specific histone modification onto that male X chromosome. It's a general modification, but it's greatly increased in its amount and location. Its location is different on the male X. So it sits on the gene bodies rather than just promoters. And then one of the more unusual aspects of this work was that there are also RNAs in this complex acting on the X chromosome, and they're called RNA on X because the genes for those RNAs are on the X, and then they also act on the X. And so the bottom line is that it turns out that this complex acts by spreading chromatin organization from the sites of those non-coding RNAs. So the RNAs themselves function in the complex and as sites, special sites. And what happens then is when the genes on the X chromosome have this complex on their gene bodies, it somehow facilitates the opening of chromatin more to allow more transcriptional elongation, which then feeds back to more expression of the genes themselves. So just to talk a little bit about this unusual aspect of it, which is that the genes for these RNAs, ROX1 and ROX2, they're on the X chromosome. So the nascent transcripts come from the X chromosome. The MSL proteins are attracted to the nascent transcripts so that their site of synthesis is a place for regulation. And then what happens after that is that they then spread in cis from the sites of synthesis. The RNAs and the proteins spread into the neighboring chromatin to create a chromatin domain. And So let me just tell you a little bit about how we found that out, which is that if you take a ROX gene for one of those RNAs, take it off the X chromosome and move it onto the wrong chromosome, an autosome, what one sees when one stains for the protein complex and the RNAs is that now, in addition to the X chromosome, now you're seeing the complex on the wrong chromosome. Not only is it going to the site of that transgene, which is very small, but it's also spreading up to a megabase into the flanking chromatin to create that domain. And so that showed that spreading from the ROX genes didn't require X chromosome sequences, that it could spread into other chromosomes if it was brought there initially. But the other thing we found from this work was that the ROX RNAs are not absolutely required in cis. That is, we still had this X chromosome with these 2 deletions for the ROX RNAs, and it still had the complex on it also. So how did that happen? We asked, well, how else might the X be marked? And so just to summarize that, basically the consensus in the field model now is that on the X chromosome, so here you may not be able to see it in blue, but this is a chromosome along its length, there are intermittent sites, which we call chromatin entry sites, that attract the complex. So in addition to the 2 ROX RNAs, there are also these special sites, and you can see these in particular mutants. Here it's MSL3 mutant. And the fact that we could get them in a mutant organism allowed us to isolate them and characterize them. And then from those sites, then, there's that spreading in cis that I told you about earlier to fill the whole chromosome, or all the genes on the chromosome. And so what we know now is that there is a sequence that's enriched on the X that we call MSL recognition element that's at these sites, and that the non-X specific part of this is the spreading is a general mark of active genes, so that once you get started on a chromosome, then you spread to an active mark on the genes to cover the whole chromosome. So that's pretty much the consensus. There's still many unanswered questions, but what's the most gratifying for us is that it's not just this system, but in different dosage compensation systems, there's this same really basic mechanism. So the components are completely different, the sequences are completely different. Actually, even the mechanisms are different in that there's repression in females instead of upregulation in males. And yet, in each case, these complexes have a very similar nucleation site, approximately 150 to 200 nucleation site, and spreading on the chromosome that should be targeted. So in C. elegans, that's dosage compensation, and also in mammalian cells with exist RNA. So what we think is that in these diverse systems, and really even beyond dosage compensation, this reveals common principles for how chromatin domains are established by nucleation sites and then can spread to fill compartments or domains. So in this part, I just want to stop now and just acknowledge some of this historical part. And obviously, Bruce Baker, my postdoc mentor, where all this work was started, and he imparted the legacy of Larry Sandler and Dan Lindsley and of genetics, taught me Drosophila genetics. Also, John Lucchese, who recently retired, who's the father of the dosage compensation field, because I didn't isolate the male-specific lethal mutants. Those came from, I stood on the shoulders of a giant there who had isolated the mutants. And Brian Turner, who discovered the acetylation, the particular chromatin modification, by looking at different modifications and mapping them to the polycomb chromosomes. And then lots of great collaborators along the way. And it's a great honor that Barry spoke before me, because actually we couldn't have cloned the first MSL gene without his help, his great mutant and his student Morris Kernan, who was working on the NAP mutant. Ron Davis, Vicki Miller, and Rick Kelly were all involved in the ROX RNAs and the spreading. Art Alexienko, more recently in the genomics and the entry sites, and Peter Park, who's an essential collaborator in bioinformatics. And I'm leaving out many, many more, but those are some of the key people. So I'm going to switch gears a little back to my original title. Chromatin analysis has taken us into some new areas, and I just wanted to share with you a discussion about how development might proceed with such specificity that we see in the animal world. And so I'm going to focus now on polycomb complexes as master switches of developmental gene regulation. So I think in biology, it's still the central question. How do we start with a pluripotent cell with just one genome? And then we go turn that into a complex multicellular organism with such reproducibility, with such specificity. And actually, I was just visiting the Museum of Natural History in Boston, and it's just unbelievable, the diversity, and yet those animals can reproducibly be made with a lot of the same genes. So how does this happen? So it's clear that, very importantly, there's cell type specific transcriptional programs. So the genome is used differently in all the different cell types. But how does that happen? So it's also clear that it happens by combinations of differentially expressed transcription factors. So those factors that have sequence specificity for the genome and turn on genes or turn off genes. Okay, but the problem comes when you look at the recognition motifs for those factors. There's a vast excess of them all over the genome. And even if you just confine yourself to looking at their binding, they don't bind all their motifs, but there's still a vast excess of binding without apparent function. So if you look at the genome, you cannot predict regulation. It's not that simple. Okay, so layered upon that is chromatin organization, and that clearly is also important. And not only histone modifications on the proteins that organize the DNA in the genome, but also factors such as the trithorax proteins and the Polycomb proteins, now all originally recognized and identified in Drosophila, but now clearly involved in development in all multicellular organisms. So with chromatin, then, you have a way of annotating the genome into active and silent. And that helps you so in the way that regulatory decisions do not have to be remade each time a cell divides. So there is a memory in the way the chromatin is organized. So that helps. So what I'm going to do now is now focus on the Polycomb group, the repressive part of this question. And there are two main Polycomb group complexes, PRC1 and PRC2, and you don't need to know most of the names of any of these, but they're highly conserved. And what they are known to do is the maintenance of silencing, and the way they do that, at least in part, is through reading and writing of repressive histone marks. So what I mean by that is reading is binding to the marks, writing is making the marks, and then, so it's a mutually, a way of maintaining a chromatin state once you've started making it. So both of these complexes are readers and writers. Sort of mutually, they reinforce each other. The main mark we'll be talking about today is H3K27 trimethyl, which is the kind of known as the canonical Polycomb repressive mark. So when you see that on genes, they're repressed. So now I'm going to take a little sideways, go a little bit away from the textbook view, because what question I'm going to ask now is stable silencing the only Polycomb role? And here's some reasons to ask that. Well, first, gene expression patterns are very dynamic during development. So if Polycomb is a kind of a silencing that once you put it on, you never take it off again, then you would be at kind of dead ends in a lot of ways with the limited number of genes we have to deal with. That clearly isn't the case. Through lineages, you see things that are Polycomb repressed, and then later in the lineage can be expressed. There can be switching. Furthermore, now that there's a lot of mapping of chromatin factors, it's clear that PRC1 is observed on active genes. Dale Dorset was one of the first people to show this in flies. It's true in flies and mammals. So we just learned it's a repressor. Why are we seeing this? And then finally, we know that Polycomb marks can coexist with active chromatin marks, and especially in embryonic stem cells. So again, this is the Polycomb silencing mark. When you see it in cells, it denotes repression. But in embryonic stem cells, on the same genes, in the same places, one sees the active mark that typically in all organisms is seen at active promoters. So I know that this histone, all the different modifications is like alphabet soup for people who don't work on this. These are the two main marks. I mean, these are the two marks I'm going to talk about for the rest of my talk. The Polycomb mark, which is repressive, and this active promoter mark. And so So what is it? How would you have these both on the same gene? We just got used to the idea that this was kind of a general conserved annotation of the genome saying one or the other. So in mammals, when Brad Bernstein and others first found this, they proposed this model, which I think is quite an attractive model. That is that in pluripotent cells, both the active and the silent mark can sit on genes. The genes are actually still repressed at that point, but maybe they're poised. Then what one sees when you differentiate these cells, for instance, in cell culture or in the animal, you see that then the genes that were poised here change and either have the active mark or the repressed mark predominant. And so it looks like you've gone from poised to a choice. And in fact, when you do that, you also see the Polycomb binding patterns and the Polycomb mark. You see that they're different in the different cell lines, that they actually, cell types. They define the cell type specific transcriptional programs. So they're defining the patterns of what genes are on, what genes are off. So that's an attractive model. But then what is the biochemical basis for that Polycomb type specific binding pattern? So true for both flies and for mammals, the Polycomb proteins are pretty much ubiquitously expressed, and they don't have sequence-specific DNA binding. So it's unclear how they go in these different patterns in different cell types, and it's just so important that they do that. That's required for the fidelity of transcription. So the patterns are different in each cell type. And so the classical model for Polycomb silenced regions is that the Polycomb proteins were attracted there by repressors or by non-coding RNAs. And so that at each gene, in each cell type, there's a specific combination of things that attract them to the right pattern. For bivalent or active chromatin regions where those active and repressive marks are together, there's not really a consensus model. So we looked at this by using cross-linking proteomics, and basically just the idea is that working on chromatin proteins is hard to do with classical biochemistry because you have to get the proteins off the chromosome, and when you do that, those are the same kind of conditions that actually break complexes apart. And so we took more of a genomic approach to proteomics by using cross-linking to keep the protein complexes together and study them. And so I don't want to go too much onto the detail because I know time is limited, but this is the work of Hyuk-Jun Kang, a postdoc in my lab, who started working on the Polycomb complexes in Drosophila embryos, and he tagged the Polycomb subunit with a dual tag of protein A and endogenous biotylation sequence that are very useful for then doing this kind of cross-linking proteomics. So in embryos, one cross-links the protein complexes to the chromatin, then purifies the chromatin and affinity purifies it using the tag. And then you can do, from the same chromatin preparation, you can do either the DNA sequencing to see what are the sequences that complex is bound to, or alternatively mass spectrometry to see what are the protein interactions that those complexes have. And so what he found, not surprisingly, was that PRC1 is as advertised. So this table basically is just showing you the top enrichment candidates. So basically number of peptides in the pull down compared to the number of peptides in an input sample. So it's truly sort of a genetic view of proteomics in the sense that the most important things should be enriched when you do this experiment. And so all the blue things came down in large numbers. But there were these other ones that were surprising because they came down in the kind of peptide numbers very similar to canonical PRC1. And yet, in fact, those are known as coactivators. They turn out to be the Drosophila BRD4, which is a very well-known coactivator in cancer research, and BR140 in Enoch, which I'll tell you about in a minute, another complex that has been implicated in cancer in mammals. So just a quick survey of this, just to tell you that BRD4 is an acetyl-lysine reader and a coactivator. So it has a Drosophila name, FS1H, and in fact, it's a trithorax group coactivator of trithorax homeotic genes known for a long time. And so it's trithorax, remember, is opposite of Polycomb in phenotype. It has the bromodomains which bind acetylated lysine, which is, again, an active mark. And it interacts with mediator and PTFB, things that activate transcription in mammalian cells and in flies. So the other two proteins are part of a four-member complex called MOS-morph in mammals. So they're homologous complexes, and we did find the other two smaller subunits farther down in the enrichment. And again, they have something you associate with activation, histone acetyltransferase domain, binding of acetylated lysines, and active marks, including that H3K4 methyl 3. And then a little bit of genetics, just go briefly through this. In fact, the genetics that has been done in these organisms from forward genetics, they've been found as trithorax mutants in flies, in zebrafish, and in fact in mouse. So what's going on here? Why would we have the cross-linking of these complexes and really abundant recovery with Polycomb of these co-activators that are transcriptional co-activators? Why would we have them together? Why would they interact? So another thing we can do with our method, as I mentioned, was to look at the binding sites where these are found. And so when June did that, he looked for this subunit of the PRC1 and this subunit of the MOS-morph. And what he found was that if you took all the Polycomb binding sites in embryos and on the genes, and it binds to promoters or transcription start sites at all these genes, and then looked for BR140, it was also there on all of those genes. All of the Polycomb genes were sites for BR140. The opposite was not true. There are many more sites for BR140. So what are those genes? Those genes turn out to be, when you look at them with gene ontology, to be all developmental and morphogenetic characteristics. And so those are for the co-bound, whereas the activator-only sites are your typical metabolism, et cetera, and your Polycomb-only sites are actually just a few terminally differentiated genes. So it looks like these factors, when we know they're together in the embryos, really abundantly together, and it looks like they're sitting on developmental promoters. So I'm just going to give you a flavor, basically, of looking at what is the status of those promoters. Now, you can't absolutely say for sure that any particular mark is on the same gene at the same time in the same embryo. But interestingly, they really fell into two clusters. They really divided into two clusters rather than a continuum. And in both of those clusters, these are chip pull-downs, just profiles of Polycomb binding to some genes, BR140 also binding to the same genes, the chromatin mark, the active chromatin mark also found on those genes. And then one cluster has a lot of enrichment of active mark, K27 acetyl, which is different from the Polycomb mark, and the BR4. So we think that group of genes are active genes that are bound with both of these factors. But the other group have abundant Polycomb repressive marks. So these are the ones with active and repressive potentially together. And so maybe it's a parallel with mammalian bivalency. It's not formally proof, but it's very tantalizing. So another tantalizing bit of information is that certainly these complexes have the capacity to recognize the bivalent marks because there's a subunit of the MOS that we isolated that does bind, has a motif that's known to bind that active mark. And then, of course, Polycomb has long been known to bind the H3K27 trimethyl mark. So if these complexes are together, they could be, in fact, binding the bivalent marks. So what is the consequence of this? So this is going to be some speculation, but some food for thought. In the classical model, there's really no reason why trithorax and Polycomb complexes would be together. In the recruitment model, first you have activators and repressors come. They determine the status of the genes in terms of activity or not. And then they attract either the activators, trithorax, or the repressors, Polycomb. So the problem with this model is that there are distinct protein-protein interactions with each gene. So it's really pretty complicated biochemically to imagine how that really happens. And in addition, there's not that much evidence for those interactions. So the field has still been struggling with how do these patterns form so specifically. So I want to suggest an alternative model, which is more of a responsive model, and that would suggest that this so-called bivalent complex, or the Polycomb and coactivator complexes, are actually sitting on all the developmental genes at their 5' ends as switches. And then what happens is when activators and repressors come into that local region at sufficient concentration, then they cause those switches then to resolve as in the bivalency model. So they can resolve either into the active states with the coactivators winning out, or in the silent state where Polycomb is increased and, in fact, PRC2 in particular comes. And, in fact, PRC2, once it spreads, may become a much more terminal mark, which may be hard to reverse. So in this model, the coactivators are stabilized by acetylation because that's what transcription factors actually do, is they bring acetylation to genes. Polycomb is stabilized by deacetylation. Repressors are known to bring deacetylases. So it's an alternative to the classical model, and I'll just then sort of summarize that in the idea that there are two states, the active state, the silent state. They're relatively stable, but if they need to be changed with transcription factors or signaling or whatever, they can be changed through reversible chromatin modification. And so this could explain why PRC1 is observed on active genes. And it could also suggest that maybe regulatory elements may never be unoccupied, and that would allow for reversibility. So, of course, we'd like to know, is this really happening in mammals? Because there's all this conservation. I have to say that it's a lot harder in mammals. Each subunit has multiple orthologs. There's a lot of change in complexes, so timing can be important for capturing key interactions. So we're struggling with this right now in terms of whether this truly is what's going on in mammals. But I wanted to finish by just saying that I think the field in general is really making progress on these fundamental questions. So starting with pluripotent cells, it's quite clear that developmental genes are bivalently marked and repressed. It's possible that when differentiation occurs that these bivalent complexes may form on bivalent promoters. That's what I've told you about today. And if so, that the master switches could then be flipped independently in response to the local activity of transcription factors. They don't have to be attracted in each specific case, but they are affected by the biochemical activity. So then, but is this chicken and egg? I mean, have we ever really gotten any farther? Because how would all of this start? Well, what's I find really fascinating is that Satoshi Namikawa and others have found that many others have found that developmental genes are pre-marked with K4 and K4, the active mark, and K27, the silent mark, in germ cells. So if that's the case, I think it's quite possible that genes may always be marked, and this might be a fundamental way that there is specificity in development. The genes don't have to be found. Everything doesn't have to be recapitulated de novo in every step. So with that, I'll just stop and remind you that Hyuk-Jun Kang did all the Polycomb work in my lab with the help of others along the way. I'm happy to take a question or two. Thanks.